community, one thing I know, I'm the only person standing between you and the people outside. So bear with me. To a person who stutters and you ask them to speed up, you are challenging the gods. Third thing, this will be a change of flavor than what the talks you have heard up till now. This is very much in the discovery phase. We do have projects which are very much in the applied phase, just like I talked before. For example, we have now a diagnostic panel on NGS, which we are now trying to put into reference laboratories in the US for the diagnosis of pediatric sarcomas. And it actually answers a lot of questions which Ian rose in his presentation is cost effective and is towards the sensitivity. And anyways, back to this project, which is the Canadian Pediatric Cancer Genome Consortium. It's all about childhood cancers. We have about 1,400 childhood cancers diagnosed in Canada. In the US or in the continental North America, that number raises to about a little above 15,000. Approximately in Canada, 200 of them they die of it, and despite progress, where everybody says that in pediatric cancers we've made progress, we are curing kids, the reality is also, as we heard previously with the platinum toxicities and things like these in children, these treatment really are very devastating. Basically, you take these kids to the brink of death and then bring them back. So again, myth and reality that we have won the war against pediatric cancer, which is not true. And again, as mentioned, even though where the cure is achieved, you leave the patient with many other things. And in the developing world, it's really a very dismal scenario. The global statistics for pediatric cancer is about a little above 230,000 cases per year. And originally from a developing country where I've kept in touch in the pediatric field, yes, the challenges are really a lot. Here is a slide which we stole from a visiting scientist from Germany. That's why it's in German. Basically, it tells you that even though everybody has been saying that the war is on, what has really happened is all your therapies in each of these categories are basically plateauing. So after you reach your plateau, nothing is happening, which means that we need new approaches, we need molecular therapies, and that's where this consortium actually came into. Again, for most of the pediatric cancers which we covered in this, the most important diagnostic or prognostic criteria is the presence of metastasis at the time of diagnosis. If you have it, this is your outcome. If you don't have it, that's your outcome. So the CPCGC was an initiative to invest high-risk childhood cancers. That also, I think it was important that they are also not part of the current other large-scale efforts. This was a $2.8 million initiative, and all the donors are up there. This included the CIHR, Genome Canada, Genome Quebec, Genome BC, and the C17 network. One of the biggest strengths of the platform, and which took a lot of effort, was to go and try to do sequencing, or what we were calling as the matched genomes. And we have single patient where we have the normal, with the primary tumor, and we have the metastatic tumor. In many cases, we also had the recurrent cases. And as we'll proceed, we'll also see that that's also what we're trying to do and capture in our validation sets. Project outcomes, what we really wanted to achieve is what are the drivers of metastatic disease. Remember, metastasis, if you have it, there's your outcome. If you don't have it, that's your outcome. Mechanisms by which somatic mutations disrupt key pathways, identification of targets for therapeutics, biomarkers, if we can have them, that would be awesome, and also especially the mechanism of clonal selection and human heterogeneity, as you heard in the earlier bioinformatic talks. With these are the benefits of Canada, we can skip in the benefit of time. The players of this project was the entire consortium was headed by Paul Sorensen, and then it also had individual leaders which were leading specific diseases. Paul is the head of the Osteosarcoma. Michael Taylor from Toronto headed the medulloblastoma, and all the people are listed. And another very interesting aspect of this project, which actually really interested me, is the GELS components and the kind of questions it asks, that what implement this kind of genetic data is going to have on society, on patients, how to handle it. For example, a question, if you do a whole genome or this kind of analysis on a kid, and you discover a mutation or something else, which is now hereditary and can affect the other brothers or sisters of the kid, do you tell the parents? When do you tell the parents? When do you tell the boy when he's older? So all these questions are arising of projects like these, and GELS is a huge component, and they will also be publishing their work. So beside the GELS component, some of the achievements of the consortium up till now, for example, efforts by Dr. Nathan Chivedo has already led to the identification of very important driver mutations that are published in Nature. Again, work emanating from this project by Dr. Cynthia Hawkins also allowed for the identification of a mutation which allows you to identify a very poor prognosis outcome or poorly performing patients. And also recently, the project headed by Dr. Michael Taylor has also been able to identify a subset in medulloblastoma. So now, osteos, which we have been dealing with, I'm on time. Good. So the CPCGC metastatic osteosarcoma is headed by Paul. A little bit of osteosarcoma usually arises from fast-growing bone planes, 2 years, or peak incidence is under 25 years of age and over 60 years of age. So it also happens in adults, but many oncologists that I've spoken to believe that what happens in all adults is totally different than when you see the pediatric population. And some of the genetic drivers which are known are also different. It's the most common childhood malignancy in cancers, and the only prognostic criteria that is available today is if you have metastasis or not. And if you have metastasis, remember the curve. So there is a bunch of things which is known about it. For example, the RB1, which is a cycle checkpoint gene, it is known to be inactivated in a percentage of these, or also P53, and also some of the on-code genes which are amplified. Again, the project aims were very in line with the aims of the overall consortium was, again, what are the recurrent mutations, what are the driver mutations, what is the mechanism of disease, clonal selection, and predicted biomarkers. So here is something very important. What we also did, we also acquired a very unique. So a lot of the effort on our end has gone into Paul's end, who has also worked very hard to assemble these patient cohorts. So now we have a validation patient cohorts in which we not only have primary tumor from them, we have met tumors from them, we have post and pre and post chemo tumors from a single patient with all these tumors. So we can really go in time and say what happens every time something is done to their tumor. So the other thing is, when we started this, we said, okay, yes, we have tumors, we have all that, we'll just go and do the sequencing. Well, it wasn't that easy, because it's how the disease is resected in surgery that you have your tumor and you have a wide resection, regardless of how careful you are of isolating your DNA. What we found is we also had the issue of we had a lot of normal DNA beside the tumor-specific DNA. And in our case, it ranged, for example, the tumor content ranged from as high to 90%, from as low as 10%. And we did our homework. These are all pathology-reviewed cases. And these are the bioinformatics outcomes. So there was also a lot of tool development over here, a very talented postdoc working on the project, Dr. Iman Lee, also in the audience. She had worked very diligently to come up with these tools to allow us to handle this kind of issues. Here are just a summary of tools used, some results. So here is a snapshot of the first 30 genomes that we did. on the basis of copy number analysis. The take-home message from this is that all over the genome, about 1/3 of the genome is all amplified arranged and things like this. So it's all over the place, the globe, okay? But also the things which were known were still coming up, and gains or losses are still coming up. For example, the magnitude of this thing also tells us in how many cases this is found to be true. So it is true that P10 is deleted in many of them. Then also patient-specific overview, but we're also finding, for example, we are finding examples in a single patient where you have massively rearranged chromosomes. So it's also coming up now as a disease in which a lot of mix and matching is happening at the genomic level. So, for example, there's chromosome number 20 from the same patient and chromosome number 8, and you can see the chromosome number 8 is all over the place. It's massively amplified, and the only way it would appear like this if there was also massive rearrangements in it. And over here, then we also have examples of very focal gains are happening. But in the end, we were also interested in recurrent primary to med. And what is emerging is that if you look at the 30 genomes and ask what is common from primary to med overall, there isn't anything which is really standing out. What is standing out that each trio or each cancer is basically a genome on its own, or it's a disease on its own. So at that point we said, okay, if this is our genomic data is showing us, can we go and see is there any other data which will help us to say, are we on the right track? So we actually were very fortunate that we have an in-house data, which was an effort out of the Department of Defense, which we were a part of, where we had 177 osteosarcoma samples, majority of them clinical primary tumors, which we already have the SNP6 analysis on it. And then we overlaid the SNP6 data with the whole genome data, and it basically overlaid beautifully. It validated almost all of our findings as far as the primary tumors is concerned. So we know, okay, we're going in the right direction. To date, because of, yes, and the time's up, I only have 2 more slides. To date, the whole genome sequencing has identified about 240 somatic coding SNVs, 63 are match-specific, 125 are shared, 147 genes across 8 genomic regions that are recurrently gained, that was big, and 219 genes across 3 genomic regions which are lost. Exomes in progress, so as I mentioned that because of the low tumor content, the way we handle it and the take-home message for it, if you are sequencing cancers, 30x, 40x, even Illumina that tells you 80x, go deeper. As much as you can spend, go and spend that money, it's worth every penny. I don't know how Genome BC feels about it. And the 200x actually is paying its dividends, and we are already seeing events which are now carrying over because we have increased our depth. So we are very happy with it. Hopefully the data will be matured in about a week. In about a month or so. For the validation, again, so what we are now doing is that we have about 30 samples in our validation cohort, which we are again doing a minimum of 200x of exome and 10x of genome. And we have in there, we had a total of about 96 samples, but 30 are going forward, which again has these very rare samples. You have pre-chemo, post-chemo, primary, MET, and multiple METs. Actually, we have 2 patients in which we have 4 different METs collected from 4 different regions. So, so those are the things that we